Welcome to BEYA Leading Voices. This engaging forum of BEYA's Leading Voices features groundbreaking discoveries, new research at the nation's historically black colleges and universities, HBCUs, and winners of the HBCU STEM Innovation Awards. Game-changing innovators at HBCUs are recognized each year at the annual BEYA STEM Conference. And now, BEYA Leading Voices.
Please welcome back to the stage Dr. Joann B. Powell.
Thank you for that kind introduction. So I am, by the purest definition, by the definition, a basic scientist. Sometimes different groups may refer to me as a biomedical researcher or toxicologist or biologist, and most often a cancer biologist, among many, many, many other things. But at the heart of it, regardless of what title is chosen in any given moment or situation, I am a basic scientist. And so tonight, I want to take my few minutes, and discuss how progress against diseases, particularly cancer, is driven by the research, discoveries, and innovations of basic scientists. As you could see from the graphic that was on my title slide, the previous slide, within a span of 5 to 10 years, up to 10,000 potential therapeutic targets can be identified by basic scientists. And we go through this meticulous process of screening and specificity studies and optimizations and preclinical testings. After all of that, we can expect 1 to 5 of those compounds to be submitted to the FDA for approval to be used in clinical trials. And we won't even talk about the percentage of those that survive the clinical trials. The graphic nicely summarized the vast amount of work that's done by thousands and thousands of basic scientists so that we can get 1 to 5 new potential therapeutic targets in a span of 5 to 10 years. And just let me mention that when we're talking about compounds specifically investigated to combat health disparities, that these numbers are even lower across the board. Basic researchers, as basic scientists, we study organisms and cells, molecules, genes, proteins, all to gain new knowledge about a cellular process that either occurs within us naturally or during the development of a disease. Basic research has broad implications because it is fundamental to our understanding and treatment of human diseases, including cancer. It's so fundamental that the NIH spends more than half of its budget, its yearly budget, supporting basic research. NIH-funded basic research projects significantly contribute to novel therapeutic identifications of new targets and to drug development. Results from any types of research can fuel this biomedical research cycle that you see here. It does that by providing observations that are relevant to the practice of medicine, which leads to questions to help us form new hypotheses that are tested in experiments in the discovery phase of research. During that discovery phase, by thousands and thousands of basic scientists, traits that are unique to a disease may be uncovered, leading to the development of a novel therapeutic. But before that therapeutic is entering clinical testing, it has to undergo preclinical testing to identify any toxicities and to help determine safe initial dosing. Clinical testing in itself is a multi-phase process aimed at demonstrating the safety and efficacy of a potential new therapeutic. If an agent is deemed safe and effective and approved by the FDA, it will enter clinical practice. Importantly, though, observations made during the routine use of a therapeutics can provide feedback into the biomedical research cycle and further enhance the use of that agent or the development of others like it. For example, an observation that African American men are least likely to achieve a PSA of less than 0.5 nanograms per mil following radiation therapy has led to new observations and launched cancer health disparity projects aimed at understanding why African American men do not respond as well to this type of therapy, or why this disparity exists at all. So what are cancer health disparities? The National Cancer Institute defines cancer health disparities as adverse differences in cancer measures such as the number of new cases, the numbers of deaths, or the burden of cancers that exist among a certain segment of the population. Cancer health disparities exist for a number of reasons that include access to health care, participation in clinical trials, which is low among the African American community, and also exposure to environmental cancer risk factors. Basic science is needed in particular if we are going to combat cancer health disparities. We need to discover new targets or methods that are beneficial to all segments of the population. The research in my lab doesn't focus on a new target, but on an old target in a new way. I mentioned that exposures to environmental cancer risk factors contribute to cancer health disparities. And if we consider that the vast majority of cancers are classified as sporadic in nature, having no familial or hereditary traits, it stands to reason that environmental toxins would contribute to cancer and contribute to cancer health disparities. My research focuses on a protein that was discovered in the 1950s and is known to mediate the harmful effects of environmental toxins, many of which we can find in our own homes, such as the chemicals that are released from fireplaces or wood-burning stoves, or the chemical fumes that are found in paints and solvents that we use each and every day, or the chemicals in our cleaning products and cigarette smokes. This protein was thought to be so dependent on a particular class of environmental toxins called dioxins that it was initially named the dioxin receptor. However, in the decades following its discovery, researchers determined that this particular protein binds to a wide range of environmental toxins, and today it is known as the aryl hydrocarbon receptor. In 2014, more than 60 years after its discovery, my lab discovered and was the first to report that AHR is constitutively active in advanced prostate cancers. In other words, in the advanced cancers, the forms in which African American are more likely to be diagnosed with, the protein no longer requires environmental chemicals for activity. It's just always on, always actively transcribing, always working. And a year after that, we demonstrated that this activity is actually sustaining androgen receptor signaling in African American prostate cancers, which of course is the driver of prostate cancer progression. Considering that advanced untreatable forms of prostate cancer is non-responsive to our current treatments that target the androgen receptor, we're in the process of investigating the effects of targeting AHR in order to control androgen receptor and reduce progression of prostate cancer to its lethal form. But we're not stopping there, because even with the current treatments available for prostate cancer, an African American man is 6 times as likely to be diagnosed with prostate cancer and twice as likely to die from this disease. With that in mind, we, and I personally feel all basic scientists, should ask the question, does our work in any way have a role in health disparities seen in prostate cancer incidence and mortality? And so we've begun to compare AHR activity in African American prostate cancer tissues. The hypothesis that we're working on was derived after reading publications from Dr. Shara that stated AHR ligands aren't just produced during manufacturing process or in our homes by everyday means, but also by frying foods at high temperatures. The hypothesis was further strengthened by Dr. Tang, whose work, along with my own personal observations and experiences, reported that soul food diets, which a large segment of the African American population consumes regularly, consists heavily of fried foods, and thus of these environmental toxins. Our investigations continue, as does the hunt by thousands of basic scientists searching for innovative targets and methods to treat and combat health disparities. So with that, my time is almost up, and I just want to end with a few thank yous, because this work is not done in isolation. Despite the reputation that basic scientists are introverted lab rats, no basic science is done in isolation. It evolves from the work of other scientists all over the world, like Dr. Leland Chung at Cedars-Sinai, who created the castration-resistant cell line that helped us confirm AHR's constitutive activity, or Dr. Puga from the University of Cincinnati Medical Center, who was the first to report that ligand-independent AHR activity even exists, or from Dr. Otaki from the University of Tokyo, who reported that AHR can indeed physically interact with the androgen receptor. So I have to thank all of these scientific warriors, known and unknown, their supporters, our social activists, survivors, colleagues like mine at the Center for Cancer Research and Therapeutic Development at Clark Atlanta, who challenge all of my ideas, as well as our administrators who always keep the trains running. And I have to thank the National Institutes of Health, particularly the National Institutes on Minority Health and Health Disparities, who recognizes that improvements in health disparities improves the overall health of our entire population. And of course, I have to thank the Department of Defense, who also champions disparities research and is funding our current project on AHR health disparities. And I finally want to thank BEYA, of course, for taking a moment to recognize basic sciences. That does not happen often. And thank you all for your attention.
Thank you for joining us for this video presentation of BEYA Leading Voices. This program was recorded at the 2019 BEYA STEM Conference held at the Marriott Boardman Park in Washington, D.C. For more on BEYA's Leading Voices, please subscribe to U.S. Black Engineer and IT Magazine or visit www.blackengineer.com. To learn about the BEYA STEM Conference, visit www.beya.org.